Okay, good. Yeah, throw out all the garbage before. Um, yeah, hi everybody. I'm Scott Pownall. I'm up in Vancouver, Canada. I'm not an automation specialist, but I love automation. I'm more a geneticist, biologist. And the later half of the talk on the open yeast collection is more my passion. But I also love open technologies, not just open biology, but also open hardware and software. And so I'm going to give you a little bit of a journey of a project that's been very much a back burner project. So it's not quite as pushed along as I'd like it to be, but partly because of everybody working on it is a volunteer. And when the main person helping out has a baby and goes on paternity leave, it throws a dent in things. But hopefully you get a gist for it. So it might not be as evolved as I'd like it to be, but we're working in that direction. Um, so let me just go next slide. Okay, so I'm going to just do a few comments about the modern commercial bioreactors. Some of you may have worked with them, some of them not, but they're basically a glass vessel. And this is like medium scale up to a larger benchtop scale rather than like the huge bioreactors or the tiny bioreactors. There's a lot of very small bioreactors that are open projects. I wanted something that was big enough to give decent cultures, but also small enough that it could fit my benchtop centrifuge. But these commercial bioreactors, they're highly flexible, so you can grow all sorts of things in it, highly configurable. Um, let's see. So the basis of it are these glass vessels. Uh, they're autoclavable, really damn expensive. Just because they've got the flange at the top and the curved bottom at the bottom, or you can get a water jacketed version where you can control the temperature by running temperature controlled water in it. That's the vessel on the right. Um, they tend to have a head plate, which is typically a grade of stainless steel that has a whole bunch of ports that you can configure. And of course, they're clamped into a tripod stand. So on the left, the head plate, you've got the top in the top left and the bottom view in the bottom right with the O-ring just to seal the bioreactor. Um, so they also come with what's this? All right, sorry about this. Getting
Important phone calls.
yeah, no, I'm just telling. I told him call after 9:15. Okay, sorry about that. As I mentioned, bioreactors are highly configurable, so there's lots of parts that you can put in, variations on the part, but they're pretty much common. So you've got septums to be able to inject things. You've got connectors to be able to add your media or whatever, depending what you want to do. You've got sampling connectors where you can actually hook up different tubes or whatever. Thermal wells for measuring temperature, baffles for controlling the vortex. You really don't want it to be, you want to make sure you're getting really good mixing. So having these baffles is important to break up the vortex from the impeller wheels. This is another form of heating, so you can pump in heat through this. Um, You could also wrap your bioreactor with like a Watt-low heating element that just sticks on the outside, and it's like a mat for heating. And this is a sparger, so if you want to pump air or oxygen in, you can do that. And of course, there are the control mechanisms for running the impeller, as well as all the measurements and things like that. Um, and I've actually been interested in having a bioreactor in the lab that's more than just a small Erlenmeyer flask that we used for culturing stuff. Um, and I came across this company, Great Glass, that they sell these borosilicate cylinders. And I wondered, well, could I 3D print a head plate, but also a foot plate to be able to create the chamber? And really, that's the core of the idea here. And One of my members of my lab, he was actually a founding director 10 years ago. Actually, this year, next month is the 10th anniversary of the creation of our lab, and in June it's the creation of our nonprofit. So, hoo-hoo, 10 years. But Casey, he works at a company where they created this, not a consumer level, but a professional level, high speed 3D printer, which is the machine on the right. So that's not a bioreactor, it's a 3D printer. His company got a grant to give like free design plus printing to us. He became a dad partway through the project, so there's a stall. He's now back kind of battling the challenges that the current Southern administration has put in place for not just Canada, but everywhere. Um, um, and so, hopefully we'll get this kicked off. I want to shout out to the Open Science Hardware Foundation. So, gosh, I got a very small grant from them that allowed me to buy the cylinders. The bulk of that grant went to my Open Yeast project, which I'll cover a bit later. Let's see. So we ended up buying two different size cylinders to give kind of the coverage that you get from the commercial. So 2 liter smaller bioreactor, and then a 9.5 liter. And the 9.5 liter was only 230 US dollars, which is a fraction of the cost of a completely sealed professional bioreactor vessel. Huge savings there. And it's a U.S. company. I went back to their website yesterday, and it looks like they were bought up. The guy that I connected to was the founding owner, and he was like in his 70s. So I guess he's passing the company on. So who knows? These prices might go up, particularly with tariffs, if he's getting the glass from China or someplace. But this is the basis of the smaller one. So it's basically a 3D printed head plate and foot plate. And so on the foot plate, we have a curvature at the bottom. And the great thing about 3D printing is you can design whatever you want. So, things that I thought about for the foot plate was rather than have a drain port that goes in, you could actually put a drain port down at the bottom of the curvature. You could also put in like swirls or ridges to facilitate more agitation and breaking up of the circular motion from the impeller. Um, but what I wanted to do is see whether the nylon itself could be autoclaved. So we used a nylon and Pantheon has a project with the Canadian military, and they're building nylon parts for medical purposes that can be sterilized, and so this was a good test of the material that they're using to see how well it's working. Um, and this is the large 9.5 liter bioreactor. And so these are kind of like the first prototypes. In the bottom right here, that's a sample of the foot plate. It was a failed print, but I used it just to make sure that it would actually fit into my benchtop autoclave. The height fits and this fits. The one above, you would mount the top to the bottom using threaded rods that would go through, and that way you'd clamp them through. But of course, you need a rubber O-ring in the bottom. Um, uh, and here I've got this is actually it's just some scrap material that I first ran through the autoclave, and it didn't deform at all. It's very stable. And then we tried threaded materials to see how well threading held up. 3D printed threading held up. So this is the M30 port, which is in the center, which is where the impeller would go. And it works really fine. Actually, just fresh printed is a bit tight to screw in, but when it autoclaves, it relaxes. And we've run these actually through the autoclave a few times now, so that we know it's pretty stable. We're getting a little bit of a warp in the flat plate, whereas this one is maintaining. And then the other thing is that because these were sitting on the bench for a long time, they did take up some moisture. But one of the challenges we have is on the base plate, of course, 3D printed, you have the ridges. And my concern was microbes, whatever you're growing in it, whether it's E. coli, yeast, or some other microbes, would it kind of get stuck within the ridges of that between cycles, and would that cause problems? So we thought about things like you can get it's like a silicone that's autoclavable. Smooth On is the company that sells it, and it's like a two-part mixture, so you can create molds. And in fact, one of my lab members, he's been working on a side project where he's using a mason jar, and he 3D printed a mold to create the head plate. And that's actually working quite well. But that's a small volume. So somebody else suggested that the inside of cans, you go and buy a can of baked beans, that they actually spray on this thermal stable coating onto the can before they put the product in and sterilize it, or pressure cook it, or whatever it is that they do with baked beans and other stuff. But I've been trying to find that. So if anybody has heard of something like that, knows a product for that, I'd love to know. The other thing is there are these bungs that you can use. So when you have a plate full of pores of ports, if you don't want to use them, you can just screw in a bunch of bungs. And we've recreated these in nylon. Let's see. So some of the other parts that we're working on, and this is my lab member Ian Caven. He's using OpenSCAD to design these impellers. And the great thing about uh, 3D printers is that you don't have to be fixed by the conventional approaches. So like if I go way back to here, you've got two different types of impellers. This is the marine impeller, and this is the Rustad. And these things are easy to manufacture in metal. But you can actually become more sophisticated in trying different designs to maximize the mixing that you get and the aeration of the microbes, depending what you're working with. Some people actually do um, uh, mammalian cells on beads within bioreactors to try to maximize things. And I imagine something like this might be a little bit harsh on those beads. And just going to go back down. And then on the right here, we've got a impeller motor, which is basically just a standard stepper motor, and then hooked up. And so Ian has developed a way to actually couple the drive shaft for the impeller onto this device. And let's see. Oh, okay. So that's kind of the bioreactor part of the thing. Oh, no, there's one more. Yeah, how did I miss that? So this is a septum. So typically you get like maybe a vaccine bottle that has a rubber septum on it, and so you need to get a injection port. And so we redesigned this as a port that can actually screw on. Um, uh, and plug it in to be able to add whatever it is that you want, whether it's adding the microbes or adding some drugs or antibiotics or whatever. So you can have a nice sterile surface that you introduce your stuff to. We've built a few other parts, but I just couldn't find the photos of them. Um, but yeah. Uh, so that's actually the finish of the bioreactor part. These are some photos I took at BioSummit. This is the 2018, which was, sorry, 2019, I think it was. So this is at Tim, I think you were there, the big party that was down there at the MIT Media Lab, and lots of fun as the unofficial official photographer for the BioSummits, as well as an organizer. Uh, and I think Derek was there as well. Okay, so I'm going to move on to the biology side of things. So I am a geneticist, not an engineer. I actually did 2 years at communication electronic engineering in Australia before I switched over to biotechnology. So I went from the hard side to the soft side, I guess. But uh, at the 2018 BioSummit, Drew Endy got up and he spoke about the open MTA and the free genes. So Drew Endy is considered one of the fathers of synthetic biology. He was at the MIT Media Lab, but moved to Stanford. He's also the founder of the BioBricks Foundation as well as iGEMS. And he got a bunch of money to actually synthesize things. So this is the BioBricks Foundation rather than Stanford. Um, uh, so synthesize DNA. This is a sponsor that funded, originally they called it the 10,000 Gene Project, but they rebranded it Free Genes. But all of these DNA materials, they've been released under the Open MTA, which was published in 2018. And Linda Call was the kind of the law guru at the BioBricks Foundation to help shepherd this through as a legal document, and it was published in October 2018. So most DNA produced in academic institution is released under the Uniform Biological Material Transfer Agreement, which means that there are barriers to what you can do with it. So you as an academic can receive it, no problems. You can work on it. You're not supposed to share it unless it's going through a hub like Addgene. Of course, academics break those rules all the time, but if they spin out a company, then they have to go back to the originating institution to sign some kind of license agreement. And the Open MTA was a legal document that was developed to permit better sharing and more opening of materials. And these are the five characteristics of the Open MTA. So access is free of any royalties or fees. You can ask for a distribution fee or things like that. Attribution, if somebody makes something, if they want attribution, then you're required to acknowledge the person that created it. Um, Reuse, so you can do whatever you want with the materials, modified or not. Then redistribution, this is the key one, is that you can actually share it with whoever that you want. And then important also is that non-discrimination. So this isn't just an academic thing. This helps a lot of startups. It helps community labs, governments, and all manner of people, because it's really unrestricted, and it's a really important tool. So with the creation of the Open MTA, which now Addgene has adopted, so if your institution is a signatory to the Open MTA, then you can submit things to Addgene under the Open MTA. If you're an academic, you got to check with your industrial liaison office to try to get them to become a signatory to the Open MTA. And there's been a growing list of institutions that are signatory. Free Genes is a library of different parts for different species. The two main ones are E. coli, and a lot of this work is from Jenny Malloy's lab at the Open Bioeconomy Lab in the University of Cambridge. But the basis of these parts are a library of parts that can be easily assembled using this technique called Golden Gate assembly. And you engineers will appreciate this. I'm a maker, but I make with DNA. The cool thing about this, so when I started, we had these classic type 2 restriction enzymes that cut DNA. So they have a recognition sequence. In the case of Hindi3 here, it's AAGCTT, which is palindromic. So on the bottom strand, it's AAGCTT. And I would spend months putting together large constructs. I used to make transgenic mice or knockout mice back in the old days before CRISPR. Um, uh, but with type 2S restriction enzymes, their recognition sequence is not palindromic. So GGTC, but if you read it backwards, it's not the same, GAACC. So that means that you can flank any sequence with a BSA1 site, for example. And the other cool thing about these enzymes is that they cut outside of their recognition sequence, unlike a Hindi3. And they have a 4 base pair overhang, which makes it easy to be able to um, uh, um, design parts and library of parts. So this is more for E. coli, but basically you have a library of promoters. These are the sequences that turn genes on and off. Then there are these ribosome binding sites that are used in microbes, prokaryotes, and archaea. You have your gene of interest, and then you have a terminator. So that's a classic transcription unit. So each of these can be a library of parts that you can put together either at the bench, or if you're doing massive scale, you can do it through automation. And basically it's a one pot assembly. So you take all your parts, stick them in a PCR tube, throw it into a PCR machine, and just run it between a couple temperatures, the optimum temperature for cutting at 37 degrees Celsius and then down to 16 degrees for T4DNA ligase. And you end up with your assembled plasmid, which has no BSA one site. So you're actually driving the assembly of this. And it's a very powerful tool. So now I'm going to lead into my project, the Open Yeast Collection. So when I heard about the Free Genes program in 2018, um, in the beginning, Free Genes was just taking individual sequences and they synthesize it for you. So you could put in a request for, a particular enzyme that you might be wanting. And they found that that was actually pretty cumbersome. So they started creating collections. So Jenny Malloy, with her group, they went in and created the coli collection. So the open enzyme collection, these are off patent enzymes that are used in biology. So there are some restriction enzymes. BSA1 didn't come off patent in those early days. Um, um, but I set about building this collection, open yeast collection for engineering yeast. And the focus is on Saccharomyces cerevisiae, as well as it used to be called Pichia pastoris, but the new name is Commigatella pfaffii. Everybody knows about this, brewer's yeast, baker's yeast, whereas Commigatella pfaffii, not a lot of people outside the biotechnology are familiar with it, but it's actually a really cool yeast that it was commercial, it was patented, the patent expired, and if you can actually take we call it a heterologous protein, so a protein that evolved in a different organism, and you stick it in here to produce the protein, but you put on a secretion tag. If you can get Commigatella pfaffii to secrete it, then that's huge with regards to purification of your protein, because Commigatella pfaffii doesn't secrete a lot of protein. So the bulk of what gets secreted is your protein. There's a lot of engineering that people have done with these. Some are still on patent, some aren't, and we're addressing those off-patent situations to maximize your protein production. Okay, so I've really gone through those two. I just want to say that Saccharomyces cerevisiae is good for metabolic engineering. So that's where you want to produce like a small molecule. So for example, the breast cancer drug Taxol was discovered in yew trees. And so they started hacking down all these yew trees to try to produce more and more, but you can't grow enough yew trees to get the supply that they need. And so they uh, um, the petrochemical industry came along and said, well, we can synthesize that. But they produced a lot of catalysts, chemical catalysts that are a toxic waste product. So the biologist came along and said, well, if a yew tree did that, maybe we can actually get adapt those genes for yeast. And so now the bulk of Taxol is produced in yeast. So it's also a very useful platform for biotechnology. And of course, the one that humans have been working with for, over 3,000 years. Recently, experiment.com had a crowdfunding challenge grant that was put on by David Kong, who was the creator of the BioSummit. He's at the MIT Media Lab. And I know Tim got his, and we ended up getting a small grant to be able to actually, for us, this is a huge grant. So this is US dollars translated into Canadian. It was even more. But we're partway through it, worrying about whether tariffs are going to chew up more of it than we want. But anyways, we'll figure that out. So the open yeast system, it's uses Golden Gate assembly, and instead of showing you the four base pair overhangs, I've just tagged them with letters. This is the classic transcription unit, and it has the same overhangs as, for example, the E. coli system. And this is the interesting part, and the rest of it is really for dealing with getting your interesting part working in yeast. And so my open yeast collection, the original, went to print back in 2021 at Twist Biosciences, and then Free started distributing, and it was also adopted by iGEMS. So Open Yeast is now in over 50 countries around the world, and there are quite a few startups that have been contacting me about working with it, both within Canada and the US, as well as Europe. Um, and I think there's a lot of interest in open tools. I won't go into the detail of all these parts, but the important thing is if you want integration, you flank your sequences with homology arms that match up with the genome that the location that you're trying to integrate in. Then there's also yeast selection markers to be able to select for cells that have taken up your construct. And these assembly connectors are for if you want to build multiple transcription unit parts. But that grant allowed us to create proper receiving vectors. The original Open Yeast collection, I had more in there, but quite a few parts failed at Twist. So it was like the bare minimum to be able to put together a receiving vector. But now, because of this grant, I created a new one, and the backbone uses there's a standard European vector architecture, and then a group in the UK created the basic SIVA vector. So I chose to go with that because basic SIVA is identical to regular SIVA, except for it doesn't have an origin to transfer, which means that if a part gets escaped out into the environment, it's not going to get transferred to microbes all that easily. Okay, this is an example of how one would assemble a construct in Pichia. And typically, it's a single gene that you're interested, so you just put in, and you just grab all these parts, throw them into a tube, throw in your enzymes, and run it overnight, and then transform that to bacteria and look for your final constructs.
Hey, Scott, quick
Uh,
time check. Got about 12 minutes.
Oh, okay. Sorry, I'm going to just rush through. So these are the pathways that you're going through, and more recently, Jenny Malloy's group got funding from University of Cambridge that allowed the creation of the Open Enzyme Collection and the Yeast Protein Expression Toolkit. This is the toolkit. I won't go into all the parts. But if you're interested in like having a fusion partner that binds to polystyrene, we have 12 different polypeptides that you can fuse to your protein. There's a whole bunch of tags for purification or other things, a whole bunch of secretion vectors. These are accessory genes to help Pichia secrete your protein. And finally, the OpenMTA is not just for DNA. So recently there's a publication about Open Pichia, and I've spoken with Katrin Clays, the first author of the paper. Their title says license free, but they give you a license and you can't share it. And they charge a fee, so not so open. Noah Sprint at Imperial College London, he did his PhD there and he started up Change Bio. I was actually working to replicate OpenPicchia, but he beat me to it. So 2 weeks ago, I had shipped to me the exact same mutant, the Hawk 1 mutant that makes Picchia easy to transform. So now I can share that with people, and I can engineer it. And yeah, it's awesome. Let's see. So yeah, gratitude. These are all the people that have helped to get this going. So maybe I'll leave it there and open the floor.
Yeah. Thanks, Scott. Let's give Scott a round.